Howdy, folks. Thanks for checking into Mr. Ulrich's LandofBiology.com. I am Mr. Ulrich. In this last of the series on macromolecules, I'm going to be talking about nucleic acids, those molecules that function to store and transmit hereditary information about how to make proteins. Now, we're going to be talking about the how and when and all of that good stuff when we get to genetics. This notescast is going to focus mainly on nucleic acids as polymers of nucleotides, and the examples, structurally the examples of DNA and RNA. To start talking about the structure of nucleic acids, we should start with their basic building blocks, which are nucleotides. Each nucleotide has three basic parts. It has a nitrogenous base, which is made up of a carbon-nitrogen ring, at least one of them, and a pentose sugar. Remember, pentose means it has 5 carbons, and these sugars have a 5-member ring as well. The sugar is either ribose in RNA, that's the R, or deoxyribose in DNA, that's the D. And it also contains a phosphate group. Remember, we met phosphates last time when we were talking about functional groups and how phosphates with all of those oxygens carry lots of negativity, and they kind of give the DNA a negative charge. There are different types of nucleotides based on the base that is associated with the nucleotide, and they break up into 2 families. First, there are the purines, which are adenine and guanine, and these have 2 rings, and the pyrimidine, which are cytosine and thymine and uracil in RNA, and these have 1 ring. We'll get to the significance of the rings here in a moment. In a single strand of DNA, then, it's really a sequence of nitrogenous base of either purines or pyrimidines that make the difference in a DNA molecule. As nucleotides are assembled into the polymer of a nucleic acid, a backbone is kind of formed, made up of the sugars bonded to the phosphates. Each nucleotide is kind of bonded together with what's called a phosphodiester bond. The new nucleotide is added to the sugar of the previous one, so the next one is going to be kind of added on top of it, and that means that the polymer grows in one direction. It's important that DNA has directionality, just the same way that it's important that a book has directionality. If it's going to make sense, it has to be read in one direction. Otherwise, it would have to all be palindromic in order to carry any kind of information. And this way, we get a sequence of nitrogenous bases kind of hanging off of a sugar phosphate backbone. By relating this ability of nucleic acids to record information as a sequence, just like a sequence of letters in a book, is a working analogy that you'll see over and over again. And DNA and RNA are not only about storing information, but DNA is the way that we pass that information on to the next generation. So in order for it to be accurate, it needs to be able to be copied very accurately. Otherwise, we'll have mistakes passed on to the next generation in the form of broken genes. Each gene is a sequence of DNA that codes for a particular polypeptide. For the rest of this video, I'm really going to be focusing on DNA. We'll leave RNA and the hows and the whys and the wheres and the whens of protein synthesis for a later video when we get into modern genetics and all of that good stuff. Here we're going to be really talking about the structural characteristics of DNA, the fact that it's a double chain, a double polymer. It's the nitrogenous bases don't just hang out there on their own. They bond to other nitrogenous bases of another complementary strand, and together the two will twist up through hydrogen bonding to make the classic double helix that we all know about. That was first developed, the structure was first developed by Watson and Crick. Of course, the unsung hero is Rosalind Franklin. She's the one who provided the X-ray crystallography. Well, she didn't really provide the X-ray crystallography data. I believe that was Maurice Wilkins who really did it. The story is interesting, and you should look into it. Well, I'll leave it at interesting. Not only can DNA hold a sequence of bases and therefore encode information, it also has to have a way to pass that information on to the next generation and therefore be copied chemically and accurately. And this is going to come about through the pairing of the nucleotides, the way that the nucleotides fit between the two backbones of our double polymer that is DNA. See, in the space, just over 1 nanometer there between the two backbones, there's really space for only 3 rings. Therefore, we're always going to get a purine and a pyrimidine. This brings us to our good friend Chargaff and Chargaff's rule. What Chargaff did is he did a lot of experiments taking the nuclei of different organisms and digesting them and taking a look at the materials that resulted from the breakdown of the nucleus. And what he recognized, he would get all this adenine and thymine and guanine and cytosine. But when he looked at the amounts, he always got the same relative amount of adenine as he did to thymine and the same relative amount of guanine as he did to cytosine. He may have had different amounts of ATs relative to GCs in different species, but he always got the same A to T, and he got the same G to C. And it turns out that the reason why there is this regularity has to do with the hydrogen bonding that connects the nitrogenous bases in their pair. Because of the way that adenine and thymine can form 2 hydrogen bonds between them, and guanine and cytosine form 3 hydrogen bonds between them, there's going to be a difference between an AT purine pyrimidine group and a GC purine pyrimidine group. So they're going to all kind of come together, and as the complementary pairs anneal, as they bond together in those hydrogen bonds, it forms this stable structure, and then we'll get some hydrogen bonding through the side chain, through the backbones, that are going to form that double helix. It's a good thing that these hydrogen bonds are relatively weak because, of course, we're going to need to unzip the DNA in order to get at the information and make a copy. Right away, as Watson and Crick figured out the structure of DNA, they realized that they had hit upon a system that lended itself to being able to make copies because of Chargaff's rule, which they knew about, that with the double helix, there would be this kind of complementary base pair system, and that means that if you have one strand, that you can automatically build the other one. And of course, this is a great system because it can be done chemically. It doesn't. It's not going to require like a person to sit down and write it down. There's a way that we can do this enzymatically, and of course, this is going to take place while the DNA is in loose stringy form, is in chromatin form, so that it is able to be unwound, just like during protein synthesis. Again, that we'll hear about later on when we talk about genetics. Well, I promised you this one would be a short one, and indeed it is. Thanks again for checking into Mr. Ulrich's Land of Biology. Again, I am Mr. Ulrich, and if you have any questions, feel free to drop me an email, put it in the comments section. Other than that, we'll see you in class.